Aging genomes and their biomarkers. There are many genetic biomarkers that are widely accepted to be associated with age-related diseases. Those analyzed in this study include GFAP, FBOX9, TREM2, and NOTCH2 genes because they correlate to longevity and diseases with age as a known risk factor. This study aims to find how changes in key genes in the genome relate to age-related illnesses.
FBOX9 is associated with the FOXO longevity pathway. Moreover, FOX proteins belong to a family of transcription factors that regulate the expression of genes associated with cell growth, proliferation, differentiation, and longevity.
GFAP is commonly seen as a biological marker for Alzheimer's and astrocytoma. It is used to differentiate between glial cells and astrocytes because it encodes intermediate filament proteins. Specifically, it is heavily involved with glioma classification.
TREM2 is a known biomarker for late onset neurodegenerative diseases. It's a gene which codes for a protein on myeloid cells such as microglia, and it's involved in immune and inflammatory responses. The overexpression of TREM2 is known to be responsible for late onset Alzheimer's due to altered activity of the microglia and their response to amyloid plaques.
The NOTCH2 gene encodes for the NOTCH2 protein, a member of the Notch signaling pathway. NOTCH2 receptors send signals that are crucial for normal development before and after birth, such as cell determination, immune system function, and tissue repair. Mutations in this gene eventually lead to Alagille syndrome, Hodge-Duchenne syndrome, and forms of cancer such as lymphoma.
In this study, we claim that the biomarkers for age-related neurodegeneration will be expressed as follows. The FBOX9 gene will be more highly expressed in adults than elders, but the GFAP, TREM2, and NOTCH2 genes will all be more highly expressed in elders than adults.
So first we utilize Open Targets platform to find genes associated with aging and disease. Then we use Expression Atlas to find expression levels of our selected genes and cross-compared the gene expression between varying age groups using the geodatabase browser. And this database was from a set of Alzheimer's patients, which we separated into adults 25 to 64, seniors 65 to 95, and centenarians 96 and up.
For the results, we have 4 graphs, one for each gene showing the gene expression in each of the groups as mentioned in the methods. In figure 1 for FBOX9 gene, higher gene expression is observed in adults compared to the elderly and centenarians. In contrast, figures 2, 3, and 4, GFAP, TREM2, and NOTCH2 genes, respectively, show a higher gene expression in the elderly and centenarians compared to the adults. Overall, based on the p-values from the data table, it can be said that all of the genes are statistically significant. The FOXO pathway is known to be associated with longevity of cells, so it is no surprise that adults express more FBOX9 gene than do the elders. Additionally, its cell type expression is oligodendrocytes, whose primary role in the CNS of certain vertebrae is to give support and insulation to exons. As such, the data supports the claim that younger individuals would express more FBOX9 than elderly individuals.
The overexpression of GFAP in elders versus adults and its statistically significant p-value is consistent with its biological late onset marker characterization and the claim of the study. It is verified that the presence of GFAP can be a determining factor in identifying age-related diseases and conditions, since it encodes for filaments of astrocytes, which are responsible for interactions in the CNS.
Likewise, elders and centenarians express a statistically significant higher amount of TREM2 than adults, which is also consistent with its known late onset biomarker identity. The higher expression of TREM2 in these patients suggests a proliferation of microglia surrounding amyloid plaques in their brains, which also suggests their further progression in the disease. In addition to neurodegenerative diseases, increased microglial populations are also associated with general aging, which makes sense for TREM2 to be a late onset biomarker.
Similarly to TREM2, the NOTCH2 gene is present in a higher abundance in the elderly patients of the Alzheimer's dataset. The data present in figure 4 and the significant p-value are consistent with our claim that NOTCH2 is expressed higher in the elderly sample populations. This is most likely due to irregular expression of NOTCH2 being linked to cancer and mutations of NOTCH2 being linked to neurodegenerative and neurovascular dysfunction. And future experiments that we could do would involve determining the effects of underexpression versus overexpression in our selected genes.
Thank you everyone for your time.